Welcome back to the Deep Dive, where we plunge into complex science and extract the most essential knowledge you need to be well-informed. Today, we are tackling a foundational challenge to human health, the principles of infectious disease.
It really is a remarkable area. I mean, it's this ancient, ongoing evolutionary battle. And as our sources highlight, we live in this era of astounding medical triumph. We have global immunization, sophisticated hygiene, powerful antimicrobials.
But there's a paradox, isn't there?
There is a huge one. Infectious diseases still make up this massive, unrelenting global health burden.
It feels like the ultimate arms race. Just when we think we've engineered a perfect defense, the unseen world adapts. Our sources today are really clear on this. Microorganisms are constantly mutating, constantly evolving, and this leads to the sudden emergence of new agents like SARS-CoV-2 or the slow, insidious creep of antimicrobial resistance, which threatens to undermine everything.
That ability to evolve is the core driver of this entire field. So our mission in this deep dive is to establish a rock-solid foundation for you. We're going to explore the biological and epidemiological principles that govern infection, focusing on that critical triad: prevention, diagnosis, and treatment.
Okay, so before we jump into the details of this microbial battlefield, we need to make sure we're all speaking the same language. The terminology here is incredibly precise, and getting it wrong can lead to, well, massive errors in clinical judgment.
Absolutely. We have to distinguish between just the mere presence of a bug and the actual harm it causes. So let's start with infection in the strict sense.
Okay.
This is when the microorganisms actually establish themselves inside host cells or tissues. They replicate, and this whole process is enough to induce a host response and cause pathological changes.
So establishment and damage, that's the key. But what if they just settle in without causing trouble?
Right. That's colonization. This is defined as the microorganisms surviving and replicating specifically on an epithelial surface, so skin, a mucosal lining, but without causing any pathological change.
No symptoms, no illness.
Exactly. It's simply coexisting. I mean, you and I are both colonized by trillions of bacteria right now.
And then there are the really sneaky ones, the agents that invade but then just go dormant.
That would be a latent infection. This is where the microorganism gets in. It survives, but it just lies dormant. It replicates minimally, if at all. Waiting for a trigger, usually a drop in the host's immunity, and then it reactivates.
Okay, so that brings us to the clinical consequence: infectious disease.
This is the tipping point. Infectious disease is when the microorganism, or, the host's reaction to it, is sufficient to cause overt illness, harm.
You see clear signs and symptoms.
You do. But, and this is a vital footnote, you can still have asymptomatic infection.
Yeah.
And that's only identifiable by actually detecting the pathogen itself or the immune response it triggered.
Now, we also need to clarify the source, because that has huge implications for public health. Communicable versus endogenous.
Yeah, a critical distinction. Communicable diseases are caused by pathogens that can be transmitted between hosts. That's your classic person-to-person spread, or, animal to person.
And endogenous infection.
They're the opposite. They're caused by organisms that are already colonizing the host. An inside job, so to speak.
And this endogenous category is a huge deal in hospitals, right? Where our own friendly flora can turn against us.
Exactly. This defines so many serious healthcare-associated infections, or HVAIs. Take MRSA, methicillin-resistant Staphylococcus aureus. It can colonize your nose or skin with no symptoms at all. But if a surgical incision or a catheter gives it an entry point,
It becomes a deadly endogenous infection.
Precisely. Or Clostridioides difficile CDI. That's an overgrowth of resident gut flora, and it's often triggered when broad-spectrum antibiotics wipe out all the competition.
It's clear that whether it's an outside threat or an internal revolt, there are necessary components for this to happen. Our sources call this the chain of infection.
That's right. The chain of infection just describes the essential elements you need for a communicable disease to be transmitted. You need the agent, a reservoir, a portal of exit, a mode of transmission, a portal of entry, and a susceptible host.
And if you break any one of those links,
You stop the spread. That's the entire philosophy behind infection prevention and control.
So let's begin by classifying the agents we're up against. In the 19th century, Robert Koch gave the scientific community a much-needed framework for defining an infectious agent.
Koch's postulates were, they were revolutionary for their time. They required the organism to be present in every single case of the disease, to be isolated in a pure culture, to cause the disease when you inoculated a susceptible host, and then you had to re-isolate it from that new host.
It created a standard for causality, a real benchmark.
It did. But today, our sources describe them as being of, well, primarily historical interest.
So why don't they hold up to modern microbiology? What's changed?
Well, the microbial world is just far more complex than Koch ever realized. For one, many crucial pathogens are uncultivable on artificial media.
Like what?
The organisms that cause syphilis or leprosy, for example, you just can't grow them in a lab dish. So that immediately violates the second postulate. And furthermore, the existence of asymptomatic colonization, which we just talked about,
Right.
that means the agent is often present in people who are not sick, which completely undermines the first postulate. So while they're conceptually powerful, they just aren't practical anymore.
Okay, so let's look at the modern taxonomy, starting with the minimalists. Viruses.
Viruses are just genetic material, either DNA or RNA, packaged inside a protective protein coat called the capsid. They aren't really alive because they're incapable of independent replication. They're what we call obligate intracellular parasites.
Meaning they have to hijack the host cell's machinery, its ribosomes, its enzymes, just to make copies of themselves.
Exactly. And the RNA viruses have a few extra tricks. You mentioned retroviruses.
Like HIV.
Right. Retroviruses like HIV carry an enzyme, reverse transcriptase, which lets them transcribe their RNA genome into DNA inside the host cell,
Which can then integrate into the host's own chromosome.
making them particularly insidious, yes.
And there's a crucial difference in their structure that determines how we contain them. The lipid envelope. Why does that envelope matter so much?
It all comes down to stability. Enveloped viruses like influenza, SARS-CoV-2, HIV, they steal a piece of the host cell membrane as they exit.
I see.
This lipid layer makes them really fragile outside the body. They're easily destroyed by detergents, heat, or just drying out, so they require close contact to spread respiratory droplets, sexual contact, blood.
And the non-enveloped ones.
They're the tough ones. Because they lack that fragile lipid layer, non-enveloped viruses like norovirus or poliovirus survive much better in the environment.
Which is why norovirus outbreaks are so notoriously difficult to contain.
Absolutely. It makes them perfect for spreading through the fecal-oral route or through really persistent respiratory aerosols.
So can you briefly trace that path of viral invasion for us?
Sure, it's a six-step dance. It begins with adsorption, where the virus attaches specifically to a host receptor. That determines which tissues it can infect.
Then what?
Second is penetration into the cell. Third, uncoating, which liberates the nucleic acid. Fourth is synthesis of new viral parts using the host's machinery. Fifth, assembly of all those new parts.
And finally, release.
And release, yeah. Either by budding off the membrane or just by lysing, disintegrating the host cell to escape.
Okay, moving up the complexity scale, we find prokaryotes, or bacteria. These can replicate on their own.
They can. They can synthesize their own proteins, their own nucleic acids, and divide independently. But they lack a nucleus. The defining feature is their rigid cell wall, which is made of peptidoglycan.
And this peptidoglycan layer is the dividing line between gram-positive and gram-negative, right?
Precisely. Gram-positive bacteria have a really thick, multi-layered peptidoglycan wall, which holds onto the crystal violet stain in the lab. Gram-negative bacteria have a much thinner wall, but it's surrounded by a second outer membrane.
And that outer membrane contains lipopolysaccharide, or LPS.
It does. And that LPS component is a massive part of their danger. It's the endotoxin we'll get to later. That double membrane also just makes gram-negatives harder for certain drugs to penetrate.
And outside of their main chromosome, they have a way to fast-track their evolution.
That's right. They use plasmids. These are small extrachromosomal rings of DNA.
And the crucial part.
The crucial part is that they can be transferred horizontally between different bacteria, even across species. This is the primary way that antimicrobial resistance genes spread so lightning fast through a population.
Okay, finally, the most complex category, eukaryotes. So fungi, protozoa, helminths.
Fungi are really important from a therapy standpoint because of their structural differences from our cells. Their cell membrane contains ergosterol instead of cholesterol, which is a great drug target.
They also have a rigid cell wall made of things like chitin and 1,3-D-glucan.
And that's the thing labs often look for in the blood to diagnose a really serious invasive fungal infection.
Exactly. It's a structural component unique to them. Protozoa and helminths, which we often just call parasites, have these incredibly complex multi-stage life cycles, often involving different hosts or environmental stages to propagate.
Okay, so we've characterized the enemies. Now for the friends. Our normal microbial flora, the microbiota, this is where it gets really complex.
The human body is a mobile ecosystem. The microbiota isn't just the microorganisms, it's their collective genes, their interactions. We now recognize it has this profound, continuous influence on our immune development, our metabolism, our overall health.
So which parts of the body are the most densely populated?
The gastrointestinal tract, especially the colon and the mouth, those are by far the most heavily colonized sites. The sheer number of bacteria in your gut outnumbers your own human cells.
What are the benefits they provide beyond just taking up space? There are three main things. They help maintain and mature our immune system. Bacteria in the lower GI synthesize essential nutrients, especially vitamin K and some B vitamins. But most critically, they provide colonization resistance against invading pathogens.
That's a fascinating concept. How do our resident organisms actually fight off invaders?
They fight on multiple fronts. They outcompete the pathogens for nutrients. They change the local environment, often by lowering the pH. They produce their own antibacterial agents like bacteriocins and fatty acids.
Wow.
And their very presence induces our body to produce cross-reactive antibodies that can then act against related pathogenic species. It's like a form of natural vaccination.
Conversely, where should we not find these organisms?
Well, places like submucosal tissues, the bloodstream, the peritoneal cavity, those are classically held to be sterile. Now, the view on sites like the lower airways and the bladder is becoming a bit more nuanced. We know some limited microbiota can exist there, but they're still heavily protected.
And this brings us back to the endogenous infection you mentioned earlier, when normal flora goes rogue.
This happens when our natural barriers get breached. The first mechanism is translocation, where organisms spread from their normal home. A classic example is a urinary tract infection, or UTI, caused by gut bacteria moving up the urethra.
And the other mechanism
Is overgrowth, like the CDI example again, where a single dangerous species just explodes in number because all its competition has been wiped out, usually by antibiotics.
Given how powerful the microbiota is, it makes sense that we've developed ways to manipulate it.
Yes. Probiotics are given to try and restore a beneficial profile, though their clinical effectiveness is still, well, it's highly debated. Depends on the strain, the disease.
But there's a more dramatic intervention.
A much more dramatic one. Fecal microflora transplantation, or FMT.
So that's the transfer of filtered gut material from a healthy person. Why does it work so incredibly well for recurrent CDI?
Because the core problem in recurrent CDI is the total loss of colonization resistance from repeated antibiotic courses. FMT instantly reestablishes a dense, diverse community of normal flora. It restores that resistance and literally starves out the C. difficile.
So it's proof of concept.
It is one of the most successful microbial interventions in modern medicine. It's definitive proof of the immense power held by our resident friends.
Okay, now we delve into the conflict itself. For an organism to cause harm, it has to get past our defenses and the colonization resistance of our normal flora. And we start by distinguishing between two major pathogen types.
Right. The distinction between primary and opportunistic is key. Primary pathogens. They have this inherent virulence. They're capable of causing disease in exposed individuals regardless of their immune status.
No, these are the heavy hitters.
The heavy hitters. Think of measles or cholera. They can take down a perfectly healthy person.
And the opportunistic pathogens.
They need a weakness. They only cause disease when host defenses are compromised. That could be a local thing, like a burn, a medical implant, an indwelling catheter.
An opportunity.
Exactly. Or it could be systemic due to chemotherapy or immunosuppressive therapy. They seize the opportunity.
So how do they actually do it? What are the determinants of virulence?
Well, they have to win at every stage. First, they have to win the competition for resources against the normal flora. They do this by sequestering essential nutrients, like iron, for instance.
Once they get past those local defenses, they have to secure their position. Which brings us to adhesion and biofilms.
Pathogens use these specialized surface molecules, adhesins, to stick to target cells. Once they're attached, many bacteria and fungi will multiply and secrete this protective matrix of sugars and proteins. This forms a biofilm.
And it's more than just a slimy layer, isn't it?
Oh, much more. Biofilms are the reason so many chronic infections persist. The antibiotic resistance within a biofilm can be thousands of times higher than for the same free-floating organism.
Why is that?
Because the matrix physically stops the antimicrobials from getting to the core. This is why infections on prosthetic joints or catheters often require the surgical removal of the device. Antibiotics alone just can't clear that microbial fortress.
Okay, next up, the biochemical weapons. Toxins. And we have to clarify the big difference between endotoxin and exotoxins.
Endotoxin is a molecule of structural destruction. It's the lipid A component of the LPS in the outer membrane of gram-negative bacteria.
And it's not actively released.
No, it is not released by the living cell. It's liberated only when the cell is damaged or dies, for instance, when antibiotics start killing them.
And its clinical impact is huge.
It is the primary driver of the devastating generalized inflammation you see in septic shock from gram-negative infections. The pathogen isn't killing the patient directly. The endotoxin triggers the host's massive runaway immune response, which then causes all the tissue damage and organ failure.
Whereas exotoxins are more like targeted biochemical weapons.
Yeah. Precisely. Exotoxins are potent proteins released by living bacteria, and they act on specific target cells or organs. Think of the highly specific effects of botulinum toxin paralyzing nerves, or the toxins from Vibrio cholerae that cause that massive, life-threatening diarrhea. They're specialized poisons.
Pathogens also ensure their long-term survival through genetic diversity.
They are masters of rapid evolution. Bacteria use those plasmids and bacteriophages to transfer virulence factors like toxin genes or resistance genes between strains. And viruses. They use their rapid reproduction and high error rates, plus the ability to swap nucleic acid to constantly generate new strains. This is why we see new pandemic strains of influenza or all the COVID-19 variants.
For an infection to become a public health threat, it has to spread. Let's outline the main transmission routes.
Right. They're categorized by the portal of exit and entry. You have respiratory spread via airborne aerosols or larger droplets. You have fecal-oral, which is ingestion of contaminated food or water. Sexually transmitted, which is direct contact between mucous membranes. Bloodborne, direct inoculation. Direct contact, mostly through damaged skin. And critically, spread via an intermediary.
So a vector or a fomite.
Exactly. A vector is an animate host, like a mosquito for malaria or even a healthcare worker for MRSA. And a fomite is an inanimate object, a door handle, a contaminated stethoscope.
This moves us into epidemiology and how we actually quantify that spread. The most famous metric these days is R0.
R0, the basic reproduction number, is a theoretical measure. It tells you the average number of new cases one infected person will generate in a totally susceptible population. So if R0 is 3, one person infects 3 others, on average.
But R0 isn't static in reality.
No, it's constantly changing. That's why we use Re. The effect of our Re takes into account the proportion of the population that's already immune, either from prior infection or from vaccination.
And all our control measures, like masks or social distancing, are aimed at pushing that Re value below 1.
Right. If Re is below 1, the epidemic will eventually die out.
And to control that Re, timing is everything. We need to know the difference between the incubation period and the period of infectivity.
The incubation period is that silent window between exposure and the first symptoms. The period of infectivity is the time during which the patient is actually shedding the pathogen and can pass it on.
And for many respiratory viruses, the infectivity starts before the symptoms.
Which makes containment incredibly challenging, yes. If a disease has a very long incubation period, like leprosy, tracking the original source becomes almost impossible.
Lastly, the patterns of disease spread.
Endemic disease is the constant presence of a disease at a baseline level in a specific area, often restricted by environment, things that need specific vectors or temperatures.
But that's changing.
It is. Climate change is rapidly expanding the geographical range of vectors, moving diseases like dengue into previously unaffected regions. When a disease suddenly increases beyond what's expected in a local area, we call that an epidemic or an outbreak.
And the ones that make headlines.
Emerging diseases are new to humans or are rapidly increasing. They're often zoonotic. They've crossed over from animals like HIV from primates or SARS-CoV-2 from bats. And a re-emerging disease is one that was previously under control but is now making a comeback, maybe because of falling vaccination rates or rising drug resistance.
Okay, when a patient presents with symptoms, we enter the detective phase. The investigation has 3 clear aims.
They are, first, confirm that an infection is actually present. Second, accurately identify the specific pathogen. And third, if it's treatable, determine its susceptibility to antimicrobial agents.
But the results are never just a simple yes or no. We have to be really cautious about interpreting results in the context of our own flora.
Absolutely. The interpretation has to consider the test's statistical performance, its sensitivity, its specificity, its predictive values, and you have to know what's normal for that site.
Right.
Finding E. coli in a urine sample is probably a true infection. Finding E. coli in a stool sample is just a Tuesday.
So let's start with the classic, microscopy for direct detection.
It's still essential. We use bright field microscopy with stains like the Gram stain to classify bacteria or the Zeal-Nielsen stain for identifying acid and alcohol-fast bacilli, which is key for diagnosing tuberculosis.
And dark field.
We use dark field for really specific but thin organisms like the spirochete that causes syphilis because it's too narrow to be seen clearly with standard staining.
But the real game changer in the last couple of decades has been nucleic acid amplification tests, or NAATs.
NAATs are the gold standard for sensitivity and speed. They detect specific DNA or RNA sequences. The key technique is PCR, which relies on exponential amplification. You start with a tiny bit of genetic material and amplify it millions of times until it's easy to detect.
And if the pathogen is an RNA virus?
Then we use RT-PCR, reverse transcriptase PCR, which first converts the RNA into DNA before amplification. And newer technologies like multiplex PCR can test for 20 different potential pathogens at the same time from a single swab.
That speed and breadth is incredible. What does the future look like?
That's whole genome sequencing, or WGS. The sources project that WGS will eventually be fast enough to do it all at once. Rapid identification, sensitivity testing, and typing, all directly from the clinical sample without needing to culture it first.
We also use direct detection to look for the organism's components, not just its genes. So antigen and toxin detection.
This is critical when time is of the essence or when the toxin itself is the main problem. The most widely used format is lateral flow immunochromatography, the rapid test.
Like a pregnancy test or the rapid COVID tests.
Exactly. A sample is applied to a strip. If the target antigen is present, it binds to antibody-coated particles, and the whole complex migrates along until it's trapped by a second line of antibodies, causing a visible color change. They're cheap, fast, and invaluable for point-of-care diagnostics for malaria or HIV.
Okay, let's talk about culture techniques. While gnats are faster, culture is still necessary. Why?
Culture, mainly for bacteria and fungi, is essential for two reasons. First, it provides a viable living organism for definitive identification. And second, it's the standard for performing antimicrobial susceptibility testing.
But the limitation is time.
A big one. Common bacteria might take 24 hours, but something like M. tuberculosis can take 2 to 6 weeks to grow.
Let's focus on the high-stakes environment of blood culture.
This is a complex, standardized process. It starts with meticulous aseptic technique during the blood draw to avoid contamination from skin flora. The blood goes into bottles with specialized medium and into automated monitoring systems.
And these systems are just watching for growth.
Continuously, usually by detecting microbial respiration products like CO2.
If a bottle flags positive, what happens next?
Speed is everything. A preliminary gram stain is done immediately and phoned through to the clinician to guide antibiotic changes. Then it moves to definitive identification. Historically, that meant tedious biochemical tests. Now labs use MALDI-OFS-MS.
That acronym alone sounds intimidating. Can you break that down for us?
Yeah, of course. Think of it as an instant microbial fingerprint scanner. A sample of the organism is ionized and vaporized, producing a unique mass spectrum of its proteins. The machine compares this unique profile against a massive database and gives you a definitive ID, often in minutes.
Which is transforming how quickly you can switch from that broad empiric therapy to targeted therapy.
It absolutely is.
Okay, moving to indirect detection, or serology. Here, we're looking for the host's reaction.
Yes, we're detecting the patient's antibody response. This is invaluable when the pathogen is hard to find directly. The key concept is seroconversion.
And that is?
The switch from a negative antibody status to a positive one. Or demonstrating a 4-fold rise in antibody titer between an acute sample and a convalescent sample taken a few weeks later.
And the type of antibody tells us about the timing of the infection.
It does. We look at the windows of diagnostic opportunity. IgM antibodies appear early, suggesting a recent acute infection. IgG antibodies follow, providing sustained protection and memory. Detecting IgG alone usually means a past infection.
And what methodologies are used for serology?
The most common is ELISA. For confirmation, especially for something like HIV, we use the highly specific immunoblot or Western blot. And for quick results, we use simple agglutination tests, which cause visible clumping.
Finally, we arrive at antimicrobial susceptibility testing, or AST. This guides the treatment.
The goal is determining if a drug concentration is sufficient to inhibit or kill the organism. We quantify this with two metrics. The minimal inhibitory concentration, or MIC, is the lowest drug concentration that stops visible growth. The minimum bactericidal concentration, or MBC, is the concentration needed to kill 99.9% of the organism.
How do labs actually perform this testing?
The standard is disc diffusion. Antibiotic-impregnated discs are placed on an agar plate that's been spread with the bacteria. The antibiotic diffuses out, and if the bacteria are susceptible, you get a clear zone of inhibition around the disc. The size of that zone tells you if it's susceptible or resistant.
We shift now from the individual patient to the population.
Hmm.
The crucial effort here is infection prevention and control, or IPC, which tries to systematically break those links in the chain of infection.
IPC is critical everywhere, but it's magnified in healthcare. The risk of getting a healthcare-associated infection, or HCAI, is stubbornly high, around 10% of patients in developed countries. And we are particularly worried about high-concern multi-resistant bacteria, MRSA, ESBLs, CPE, and GRE. These are organisms that render our frontline drugs useless.
Let's start with the cornerstone of IPC.
That is simply good hand hygiene. Decontamination before and after every patient contact. However, there is a critical detail here involving spores. Alcohol gel is great against most bacteria and enveloped viruses, but it is completely ineffective against spore-forming pathogens, most notably C. difficile.
So when a patient has confirmed C. difficile diarrhea,
Hand washing with soap and water, followed by complete drying, is mandatory. The friction is needed to physically remove the spores. Relying on alcohol gel during a CDI outbreak is a fundamental and dangerous error.
beyond hands, we use isolation precautions. There are three main categories.
First, airborne precautions. These are for pathogens that remain suspended as small aerosols over long distances, like measles or TB. This requires a specialized negative pressure room, and staff must wear fitted N95 masks.
Second, droplet precautions.
These handle larger respiratory droplets that typically fall out of the air within a meter or so. Think influenza or mumps. This requires surgical masks and spatial separation.
And third, contact precautions.
This is for infections spread by direct contact or via fomites like C. difficile or MRSA. It requires staff to wear gowns and gloves. And of course, the recent SARS-CoV-2 pandemic required layers of all of these depending on the context.
Scaling up, we look at outbreak management. Let's quickly define that hierarchy of spread again.
A cluster is just an aggregation of cases. An outbreak or epidemic is an excess of cases above the expected rate in a local area. And a pandemic is a massive, widespread epidemic affecting multiple continents.
When an outbreak is suspected, what are the first steps the public health detectives take?
First is confirmation. You need lab evidence, often through genetic typing, that the organisms are identical. Then you need a clear case definition. Who counts as a case? Then epidemiologists perform active case finding, often plotting an outbreak curve to visualize the infection rate over time.
And how do they figure out the source?
Through case control studies, they interview affected individuals and unaffected individuals to find a statistically relevant source. Was it a specific food, a contaminated water supply, or just person-to-person spread?
The sources detail the extreme nature of a pandemic response, something we all experienced firsthand.
A pandemic requires extreme non-pharmaceutical interventions, or NPIs. Things like mandatory travel restrictions, entry screening, isolation, social distancing, lockdowns, closure of public transport. It's all about pushing that effective R, the RE, below 1, regardless of the massive social and economic cost.
Okay, next, let's move to immunization strategies. Let's contrast passive and active protection.
Passive immunization is the transfer of preformed, targeted antibodies. The advantage is immediate protection. The disadvantage is that it's short-lived. We use this for post-exposure prophylaxis against things like rabies or tetanus, where you need defense now.
And active immunization, or vaccination, is the long game.
This is about stimulating the host's own immune system to generate memory. This can involve administering whole organisms, either leave attenuated, which are weakened, or inactivated, killed, or just specific components of the organism.
And there's a really vital distinction in component vaccines between simple polysaccharide vaccines and conjugate vaccines.
This is a huge clinical insight. Simple polysaccharide vaccines, like the older pneumococcal vaccine, they only activate B lymphocytes. This gives you a short-lived antibody response with no long-lasting memory. And infants under 2 can't respond to them at all.
So what was the breakthrough?
The conjugate vaccine. By chemically linking the polysaccharide to a protein carrier, the vaccine becomes visible to T lymphocytes. T-cell activation gives you a stronger, sustained memory-generating response that lasts for years. This innovation was literally life-saving for protecting kids from meningitis and pneumonia.
Finally, the newest tools: nucleic acid and viral vector vaccines.
Nucleic acid vaccines, like mRNA vaccines, they encode the microbial component. When they're injected, they make your own cells become temporary antigen factories, stimulating a powerful immune response.
And viral vector vaccines.
They use a modified, harmless virus to deliver the genetic instructions for the target antigen. Both of these technologies were central to the global COVID-19 response.
The ultimate success story of all these strategies is herd immunity.
Herd immunity is achieved when enough of the population is immune that the entire population, including those who can't be vaccinated, is protected. The pathogen just runs into too many dead ends.
And it works.
It works. The global eradication of naturally acquired smallpox proves it, as does the steep decline in wild polio cases worldwide.
Let's turn to the strategy for treatment. The overarching philosophy here is antimicrobial stewardship, or AMS.
AMS is a structured system designed to optimize antimicrobial use. The goal is twofold: improve patient outcomes and reduce the selection pressure that drives antimicrobial resistance, or AMR. Our sources are clear that AMS and IPC are fully complementary. Good IPC reduces the need for antibiotics, and good AMS reduces the reservoir of resistant organisms.
The roadmap for therapy is called Start Smart, Then Focus.
It perfectly describes the clinical imperative. In severe infections like sepsis or meningitis, you can't wait for lab results. Empiric therapy, Start Smart, means immediately starting broad-spectrum antibiotics aimed at the most likely pathogens. It's a life-saving intervention.
But this smart start comes at a cost.
It does. Broad-spectrum agents inevitably disrupt the normal flora and exert maximum selection pressure for resistance. That's why you have to quickly move to targeted directed therapy, then focus. As soon as the lab provides the culture and susceptibility results, you de-escalate. You switch to the narrowest spectrum agent that works. This minimizes collateral damage.
The focus on AMR is paramount. Let's detail the microbial counter-strategy, the mechanisms of resistance.
Resistance is either intrinsic, just an inherent property of the organism, or acquired, usually via mutation or the horizontal transfer of resistance genes on things like plasmids.
What are the main ways they physically defeat the drugs?
Number one is enzymatic degradation. Bacteria produce enzymes that just break down the drug molecule. The classic examples are beta-lactamases like ESBLs and the feared carbapenemases. They destroy our most potent frontline drugs.
Hey, what else?
Target modification. The microbe changes the part of its structure the drug targets. The best example is MRSA, which alters its penicillin-binding protein so the drug can no longer bind. Then you have active efflux, where the bacterium installs pumps to actively eject the antibiotic. And finally, impermeability, where it just blocks the drug's entry.
This race is leading us toward the post-antibiotic era.
We are seeing a steady, inexorable global progression of resistance. It's terrifying. We're increasingly forced to rely on older, more toxic, more expensive drugs. And this crisis is fueled by inappropriate use-giving antibiotics for viruses, underdosing, massive use in agriculture. The emergence of multi-drug-resistant and extremely drug-resistant tuberculosis shows the stark reality. Infections we thought we'd controlled are becoming untreatable.
To make sure the drugs we have work, we need to understand pharmacology, starting with pharmacokinetics, or PK, what the body does to the drug.
PK covers absorption, distribution, metabolism, excretion. Clinically, this determines how we give the drug. For instance, in sepsis, gut absorption is impaired, which is why we have to start therapy intravenously. Distribution is also vital. Gentamicin doesn't get into the CSF well, so it's useless for meningitis, even if the bug is sensitive in the lab.
And the reverse. Pharmacodynamics, or PD, what the drug does to the bug, this dictates our entire dosing strategy.
PD divides drugs based on how they kill. First, we have concentration-dependent killing. Here, efficacy is maximized when the peak concentration, the Cmax, is way above the MIC. These drugs, like aminoglycosides, have a strong post-antibiotic effect.
Meaning they keep killing even after the drug concentration drops.
Exactly. So clinically, that means a single high dose optimizes killing while giving the body time to clear the drug, which actually minimizes toxicity.
And the other group?
Is time-dependent killing. Efficacy here relies on keeping the drug concentration above the MIC for as long as possible.
So that requires a different approach.
It requires frequent dosing, or ideally, continuous infusion. Beta-lactams and vancomycin work this way. A continuous infusion in the ICU maximizes that time above MIC and is often a superior clinical approach for severe infections.
For drugs with narrow therapeutic windows, we have to monitor levels with therapeutic drug monitoring, or TDM.
TDM is non-negotiable for drugs where the effective dose is close to the toxic dose. Vancomycin and aminoglycosides require TDM to make sure we're at bactericidal levels without causing kidney or ear damage.
Let's quickly review the major specific drug classes, focusing on safety and scope, starting with the most used group, beta-lactams.
These are bactericidal. They inhibit cell wall synthesis. A huge clinical challenge is allergy. Up to 90% of patients who report a penicillin allergy do not have a true life-threatening IgE-mediated allergy, and that label often forces clinicians to use unnecessarily broad-spectrum drugs.
Next, fluoroquinolones, like ciprofloxacin.
Broad spectrum, excellent tissue penetration, but crucial safety warnings here. Risk of tendinitis and Achilles tendon rupture, especially in older patients. They also have CNS effects like delirium and can cause serious cardiac arrhythmias. They are powerful but must be used very carefully.
What about the crucial reserve agents for resistant gram-positive bugs?
The glycopeptides vancomycin and teicoplanin. Gram-positive only, reserved for MRSA and resistant enterococci. If you infuse vancomycin too quickly, it causes red man syndrome, which is a histamine-mediated rash, not a true allergy. You just slow the infusion.
And what's the one thing we absolutely have to know about oxazolidinones like linezolid?
Linezolid is another critical agent for resistant gram-positives. The non-negotiable safety point is that it has potent MAOI activity. It must never be co-administered with other MAOIs or SSRIs because of the risk of precipitating a potentially fatal serotonin syndrome.
Finally, a brief look at antivirals.
Most viral infections resolve on their own. But for specific threats, we have targeted drugs. Acyclovir is for herpes viruses. For influenza, oseltamivir, or Tamiflu, is a neuraminidase inhibitor. It has to be started within 48 hours of symptoms to be effective. And the SARS-CoV-2 pandemic brought us remdesivir, an IV drug that has shown benefit in decreasing time to recovery, especially when used early.
We have executed a monumental deep dive today. We've covered the evolutionary mechanics of pathogens, the structural differences that allow us to target them, the sophisticated techniques we use to diagnose them, and the strategic principles for public health control and treatment.
We've seen the sheer power of IPC measures, hand hygiene and isolation, in breaking that chain of transmission. And we finished with the complex territory of AMS and targeted therapy, that constant tightrope walk between saving a life now and preserving our antimicrobial arsenal for the future.
And the ultimate challenge remains the adaptability of these microorganisms and the looming shadow of antimicrobial resistance.
It is unavoidable. Any antimicrobial use, however justified, exerts a selection pressure. Even when we use combination therapy to prevent resistance in the pathogen we're targeting, say MDR-TB, those drugs still affect the vast population of commensal bacteria that colonize us.
Which inevitably selects for resistance in our normal flora as well.
It does.
And that takes us full circle right back to the start of our conversation, the concept of endogenous infection, where our own beneficial flora causes disease.
The profound irony of modern medicine is this. We rely on broad-spectrum empiric therapy to save lives in crisis. But this very practice, this necessary disruption of our gut and mucosal microbiota, contributes to transforming those friendly, colonizing bacteria, which were once our allies,
into the next generation of highly resistant opportunistic pathogens. So we leave you with this final provocative thought. Given the constant selection pressure we exert, how much are our own life-saving treatments inadvertently contributing to weaponizing the most intimate members of our human microbiota?
It's the ultimate ecological trade-off, and one that requires constant vigilance and innovation if we are to win this microbial war.
A vital truth to chew on. Thank you for diving deep with us today.